Welcome to one more session on BTOE5, viruses, friends or foes. In this last unit, we are going to learn about how to develop virus resistance in case of the plants. So if it is an animal or a human being, you're going to, if these diseases are there, you're going to use some antivirals or sort of a vaccine to protect against the viruses. Now, when you're talking about the plants, how do you create the resistance of the virus infection in the plants? So those plants, we term them as virus-resistant transgenic plants. First, let us talk about what is transgenics. Transgenics are the crops which are also called as genetically modified crops. Trans means from different organism. So a transgenic plant basically contains a gene from another organism which have been artificially inserted, or the plant acquires it through the pollination or natural process. So this type of transgenesis leads to the advanced trait of a plant. We are talking in our case the genes which can give resistance to the virus infections. Now, how do you create a transgenic plant basically? So in order to carry a gene of interest through a plasmid, again, as I have told in many of my other sessions, previous sessions, you need one mode of transfer. So the mode of transfer in the case of the plants is through a bacterium called as Agrobacterium tumefaciens. So this is a natural genetic engineer, and the nature of this bacterium is that it causes a disease called as crown gall disease. If a natural a natural isolate of the Agrobacterium infects a plant, you can see this is the disease that will happen. This is called as crown gall disease. So there are various strains of Agrobacterium like LBA4404, you have GV3101, we have EHA105. There are many different strains of Agrobacterium that are being used. But you see that this organism comprises of lot many genes which can give the lethality. So what we are going to do is we are going to develop an Agrobacterium vector or a plasmid, we call it as. It's a self-replicating double-stranded form of DNA. So to carry our gene of interest into the plant. So there are two species of Agrobacterium. One is tumefaciens, other one is called as rhizogenes. One causes this crown gall disease, what I shown you. The second one causes the hairy root disease. That is Agrobacterium rhizogenes causes the hairy root disease. So there are two types of plasmids. In case of tumefaciens, you have a plasmid called as Ti plasmid. In case of the rhizogenes, you have plasmid called as Ri plasmid. Ti stands for tumor-inducing plasmid. Ri stands for root-inducing plasmid. That is the hairy root culture producing plasmid. Let us talk about the Ti plasmid because we are going to use only tumefaciens to transfer our gene of interest. So this is called as the left border and this is called as the right border of the plasmid. So whatever the DNA is in between this region is called as tDNA, and this part will be integrated into the plant. So there have been naturally auxin, cytokinin, and opine producing genes, which we'll take out and we'll insert our gene of interest to send it to a plant or to transfer it to a plant. So this is how the general strategy of transgenic plants happens. So you have a transgenic gene or a target gene which you want to transfer into the plant. You have to clone it into a binary vector or an Agrobacterium vector which can take this particular target gene. So I can cut this with a set of enzymes called as restriction enzymes. We can clone this gene into that plasmid. Once the gene is cloned into the plasmid, we're going to place back this plasmid back into the Agrobacterium. You can see here. So this is the bacterium. And this bacterium, when you are infecting the plant, how does this infect the plant basically? The source for the Agrobacterium to move in is a wound. You have to wound the plant tissue into which you want to send your transgenic gene. So we'll wound a plant cell so that the plant will produce certain phenolics through which the Agrobacterium is attracted towards it. And that Agrobacterium will transfer your gene of interest from the tDNA into the plant genome. So this DNA integrates into the plant DNA and remains as an integrated plasmid like this. Okay. So once the transformation is done, the next step is you have to select only the plant tissues which have your transgene. For that, you have something called a selection, where you use an antibiotic, and that antibiotic resistance gene is present on your Agrobacterium vector already. Like you can see here, This is an agrobacterium vector which consists of a kanamycin resistance gene. So if you use kanamycin antibiotic in your plate, where you will put all your plant tissues which are transformed with agrobacterium, only those plant cells which have the transgene that is having this antibiotic resistance will germinate, others will die. So this germinated transgenic plant will now produce your foreign DNA along with the kanamycin antibiotic resistant gene. Okay. Now, this is how a transgenic plant is produced basically. There is something called as even a reporter gene, what people use, whether to know if your transgene is there here. How will you ensure that the transgene is expressing in the plant? That is, the transgene product is producing in the plant so that it will give the virus resistance. For that, downstream of your existing DNA, downstream means after the DNA, you're going to clone one reporter gene. For example, in our case, let us consider this reporter gene. It's called as GFP, green fluorescent protein. It is isolated from a jellyfish called as Aquaria victoria. So these are the different types of green fluorescent proteins. When they are introduced into various organisms, you can see here in rabbit, in case of the fly, in case of the Ascaris lumbricoides, worm, as well as in case of fish, this gene has a property to glow. That is, whatever the transgenic plant you have produced, if you are putting it under UV light, it will show fluorescence if your transgenic gene is present. That means you are going to engineer that gene plasmid with your GFP gene. So along with your foreign gene, that is your target gene, you have an antibiotic resistance gene and a reporter gene. Okay. Now, these are the different variants. You can see the plants here, tobacco plants, which are showing the GFP expression. You can see here, this is the tobacco plant showing the GFP expression. That is, whatever the transgenic plant will glow in dark like that. Okay. Right. So this is one of the strategy how people produce from the scratch a transgenic plant. Let us look into an example of tobacco here. So people will take leaf pieces of tobacco like this, they'll put into the plant tissue culture media, and then they infect with the agrobacterium. Once they infect with the agrobacterium, then they will select only the tissues with the transgenic plasmid or your transgenic gene, that is through the antibiotic selection. Those selected transformants are then regenerated on the plant tissue culture medium. So plant tissue culture media consists of the nutrients that are essential for the plant to grow. This is not a different one. It's a common media. So the plant tissues grow up, and when the plant tissues mature, they are also rooted and they are transferred into the potted plants. Okay, so this is how people do the transgenesis. This is example of one of the plants. I hope this is a maize plant. And this right panel is a tobacco plant. You can see here. Okay, there are many transgenic. This is one of the examples how people produce a transgenic rice. They'll start with something called as callus, undifferentiated mass of cells from the plant tissue. They'll transform the callus, select the only to the transformed callus or the ones with the transgene, and then they will regenerate the plant from this callus. Once they regenerate the plant, the final transgenic plant is obtained. Okay. Now let us come to the story of virus-resistant transgenic plants. How we are going to do that? The first example, the first ever produced virus-resistant transgenic plant was for against a virus called as tobacco mosaic virus. Remember, this is a very important question in the examination point of view. The first transgenic plant produced against the viruses was for the TMV, tobacco mosaic virus, which is a very old studied virus. So this viral genome has many genes. Among them, one gene is there which will produce the transgenic code, which will produce the code protein. Let me show you. This is the code protein gene. So this code protein gene is amplified by techniques like PCR, and they are cloned under a viral promoter so that this gene is expressed inside the plant. So when they have cloned this code protein gene into the transgenic binary vector construct or the plasmid, they will transfer this particular plasmid using the agglomerated transformation. Again, they will do the same procedure which I explained in the previous slide. Like this, they'll regenerate the whole plant, tobacco plant. So once they have regenerated the tobacco plant, when this tobacco plant was inoculated with the virus, that is challenged with the virus, when virus infection is given to this plant, it was resistant to the tobacco mosaic virus infection. How come? Here is the mechanism. What happens is that if you see the code protein of the tobacco mosaic virus, okay, so let me show you. This is the protein of the tobacco mosaic virus, which forms the virion or the capsid. When these tobacco mosaic virus code protein particles are expressed inside the plant cell, they assemble themselves like this into pseudo or empty capsids. Why these are called as empty capsids? They doesn't have the RNA. That is the full viral RNA is not there, but still they have the coat protein gene. So they will assemble themselves into the empty TMV particles or pseudo tobacco mosaic virus particles, something like this. Empty particles. The empty rods will be produced, as we have seen in case of VLPs, virus-like particles. So when these empty particles are there in the cytoplasm of the host cell, suppose a TMV natural virus comes and infects the same host cell with these empty virus particles. What happens? During the replication, tobacco mosaic virus has to release its RNA into the host cell, which has already these empty TMV particles. Once they detect this TMV RNA coming in, these empty particles will go and encapsulate. That is, they'll act like a capsule. They'll cover the TMV RNA so that the TMV RNA would not be able to replicate further, because these empty capsids will literally swallow or they will encapsulate the TMV RNA. So this is how virus-resistant transgenic plants are produced for a tobacco mosaic virus. Again, to review, what they have done is they have taken the coat protein gene, they expressed it in the plant. When they expressed it in the plant, it produces tobacco mosaic virus coat protein, which assembled into empty virus particles or the virions or the capsids. When a virus RNA comes in, these empty virions actually encapsulate the tobacco mosaic virus RNA, thereby inactivating the RNA replication. Similar type of strategy was used for one more virus called as ZYMV, zucchini yellow mosaic virus. So which actually infects the zucchini plant. Zucchini is nothing but equivalent to our, the kira or the cucumber which we consume. It's a sort of the type of cucumber plant, basically belonging to cucurbitaceae. It's not cucumber, it belongs to cucurbitaceae. And they have seen that, you can see here, that the transgenic plant expressing the coat protein of ZYMV was able to withtolerate the infection. When its control, which doesn't have the transgene, was highly infected with the virus. You can see the difference between these two images. This is transgenic plant. This is the natural plant which is infected with the ZYMV infection. So in India, if you want to produce this type of transgenics, you can't do it very easily because there are many regulatory authorities from where you need to get the permissions. What are those? There are certain authorities I want to make you aware of those things. One is called as RCGM, the Review Committee on Genetic Manipulation of the Crops. RCGM. It comes under DBT, Department of Biotechnology, Government of India. One more is there called as GEAC, that is Genetic Engineering Appraisal Committee. This committee will basically decide whether your transgenics can go into the environment. This comes under the Ministry of Environment and Forests and the Climatic Change in India. Okay. So these are guided by an institutional biosafety committee, which will decide, your institute will decide, that biosafety committee will decide whether you can do the transgenic work or not. Okay, this is the GEAE website, GEAC website, and this is the RCGM Secretariat at Department of Biotechnology, Government of India. With this, I hope you have understood how a virus-resistant transgenic plant can be produced. And the TMV example is very good core example to understand how a transgenic plant could be produced. I hope you understood how we can engineer resistance in the plants for the viral infections.